Hi, everyone. I'd like to welcome you to today's panel's discussion, Paving the Way to Precision Medicine, a Single-Cell Sequencing Perspective. My name is Kevin Taylor, and I'm in the Proteogenomics group at Revvity, and I'll be your moderator for today's event. The panel discussion is hosted by LabRoots and brought to you by Revvity. To learn more, please visit revvity.com. We encourage you to participate today by submitting any questions you may have during the discussion. All you need to do is type them into the Ask a Question box and then just click Send. We'll answer as many questions as we have time for at the end of the discussion. Also, if you experience any technical issues, you can use this same QA box and we can assist you directly. So I'd like to welcome our panelists today. First one is Nick Matthews, head of Imperial Genomics Facility, Imperial College London, UK. So Nick, thank you. Linda Orzolek, head of Single Cell and Spatial Transcriptomics Lab Operations at OMAPX. Welcome, Linda. And Dino Carlo, professor and chair of the Department of Bioengineering, University of California, Los Angeles, and founder of Partillion. So thank you and welcome. Without further ado, let's just jump in and start the panel discussion. And I think maybe we just, whoever would like to answer this first, but just to kick off some basic questions. In your experience, how are people using single cell today?
Happy to jump in. Go ahead, Nick. Sorry.
Well, on the whole, it's dependent on if you see single cell as spatial single cell or single cell as single cell with no resolution in the spatial environment. I think for single cell, we're seeing a lot of people using that for research, and in spatial, there's a movement over into pathology. I think that's the simplest answer.
Great. Can I ask you sort of what are your current interests in sort of single cell and spatial, and maybe a little bit about the path of how you got to what you're working on today?
Is that to me?
Yes.
Okay.
And then I'll ask it to everybody.
Okay. What was the first part again? Sorry.
Can you describe a little bit about how you first got into single cell and spatial, and sort of where your focus is today?
Well, my first endeavor into single cell was when I was at CRUK LRI in 2010, all plate-based with Malbac. I'm not sure if anyone remembers that, but it's all DNA-based. And that was my first endeavor into the single cell side, really trying to level out the playing field in terms of the data. When I moved to ICR, Institute of Cancer Research in Chelsea, that changed. We were quite early adopters of the 10x controller or 10x system for single cell, and that was mostly based around breast cancer, actually, and research based in that. And I think that's been where we started looking at the spatial environment, and initially with 10x, and that's expanded into other areas like, I suppose it's Bruker now, it's not nanostring, and stereomics and Curio.
Great, thank you. How about you, Linda?
So I'm not quite that extensive back in 2010, but I started with the single cell in 2017. My career is based around service. So I was part of the single cell core at Johns Hopkins, and then moving now into lab operations at OMAPX, where it's a focus on, providing access to these technologies to smaller labs. So certainly at Hopkins, tremendous amount of research starting really in the neuroscience field in 2017 with the 10x. We were at V2 when we first started, but also bringing in SplitSeq before it became Parse. And rapid increase in that interest in 2020, right after the pandemic, when I think prices started becoming a little bit more affordable with access to the NovaSeq. We started to see that increase in the single cell usage. We moved into spatial with the immediate release of Visium, and then we're also operating with the Xenium in situ platform. And with now, current focus is really on exactly what we're kind of here to talk about, is that precision medicine, seeing the movement from research to clinical applications, with our particular passion being, and my particular passion being on the cancer side, but with a tremendous outreach into the neuroscience field as well. It has been kind of where our experience has led.
Thank you. How about you, Dino? Sort of your path and what you're working on today?
Definitely, yeah. So I am a tool developer. So I've been developing tools in the single cell space before people were even thinking about, what they're asking the questions, why should I even analyze single cells? They're like, I can just do a Western, or I can do a gel and mash up a bunch of cells all together. And so in the about 20 years ago, we were making tools to analyze the functions of single cells, so enzymatic functions and other things at scale using microfluidic technologies. Over the years, I've developed a number of technologies looking at function of cells. So that can be a variety of different functions, whether it's how contractile cells are, how adhesive they are, how much they secrete, and how they interact with other cells. And so that's what we've been really focused on, is how can we bring that type of functional layer to single cell transcriptomics. And so we've been integrating our technologies through our work, which we call lab on a particle work at Partillion Bioscience in particular, with the traditional single cell transcriptomic and sequencing technologies, to add that functional layer on top and really annotate. You're seeing all this amazing data, but it's hard to make sense of it. I think that's where everyone is right now. Is like, you have all this data, what the hell does it mean? And so that functional layer, I think we think that that's going to be really critical to start to make sense of this data and use it, whether it's in clinical applications. An area that we work in also is in characterizing, for example, T cell receptor repertoires, but laying that functional information on top, so that now you have, okay, how much cytokine did that particular T cell secrete, and what was its TCR, T cell receptor, and what was its transcriptome? So all that layered together, I think, gives you a fuller picture of what these cells are doing, and allows us to use this information in a much more effective way.
Thanks. Yeah. So one of the, I guess, interesting things to me about both these fields is, I got into it in sort of late 2015, early 2016. But when you think about what are the, moderators of these technologies, right? And it's, the number of cells that you can analyze, working with challenging samples, dealing with sparse data, dealing with large data sets. And if I, look back over the past 5 years, we can do more cells, we can do them easier, there's expanded applications, and there's better analysis tools. Yet when you sit down and talk to people, it's still the need to potentially do more cells, the need to make it easier to expand to more sample types, as well as making data analysis easier. So it's still the same problem. We've just gotten better, but still need to get better. So with that in mind, for the projects you have and the people that you work with, what are the biggest sort of economic and technical challenges that you're encountering today? And if it's okay, I'd like to start with you, Linda.
Yeah, absolutely. And that's been one of my focuses over the years is really, coming from academia, it's all about education and it's all about, getting people to understand exactly the things that you're talking about. Where are the limitations that single cell and spatial presents? And how do you take a lab that's not an expert in these technologies and enable their research? And that's why, I think that the cores and the CROs are such an important part in this research. I worked for years at Hopkins and processing, to the point that 90% of what we were doing was single cell and spatial. And the number of publications that come out of Hopkins is incredibly high, but the number that are focused on single cell was so low. And it was always that impediment with data analysis and understanding the complexity that goes into it and being prepared for it. And a lot of people weren't. They really walked into it expecting that this is going to be similar to what we've developed from microarrays and bulk sequencing, so that you'd have people who are spending years, if not more, on these data sets because they think it's something that should be simple and direct. So it's the first challenge that I always want to address with anybody who comes forward is how are you planning on doing your data analysis? What pipelines do you have in play? What teams are you working with to do that? And I think that's the most daunting question that has to be addressed first. On the other side of things, it's understanding how to properly preserve your samples and prepare them so you can get them to a place where they're ready to be processed. For people who have, a Chromium controller in their lab or have are comfortable doing a, combinatorial barcoding, it can be relatively straightforward, but the vast majority of research labs are not working with that, or to get the throughput they need or the price points that they need, it becomes really, really difficult. So understanding ways to just properly collect and store and prepare samples is always, to me, the biggest conversation that needs to be held. If you're doing cryopreservation, are you comfortable with the concept of losing delicate cell types? How is that going to impact the data? What percent of cell dropout are you going to see simply because of the way that you approached collecting those samples? We get patient samples coming into the OR at 2 o'clock in the morning that you got to grab a tissue section from and be prepared to process. Most people, while we all know on the research side that you're going to end up in the lab at all times of the night and day, you have to make sure that whoever's doing the processing of those samples is also going to be there to support that. So there are these underlying factors that go beyond choosing the technology that you're going to use. Do you need 10, 20,000 cells to be able to identify all of the subclusters within your sample set? Or, is being able to do a couple hundred cells, but in a more complete fashion, being able to identify more genes within that going to be beneficial to you? They're very, very different questions. And it means that these projects are never as straightforward as people, walk into your office and assume that they're going to be. I've always made it the policy of, you don't show up with samples. You have to go through all of this pre-processing discussion to produce a successful result. So the number of, I've done entire presentations just based on all of the questions that need to be asked. So that list goes on, and we certainly, it's almost to the point where you can't address all of them because of what comes up during the processing. But doing those bookends on either side between understanding the data analysis complexity and working with people who have that experience versus, the prep work that goes into designing the project are really the two most important steps. I always joke and say, anybody can do the lab work. I've learned over the years that that's really not quite true. And you want to make sure that you have those expert hands doing it. But if you don't address the bookends first, it doesn't matter what happens in between.
Definitely. Thank you. Yeah. Generally in my lab, when it comes to single cell or spatial, just sort of intuitively when we're bringing somebody up, to speed, it's, it's 4 months at least, right? And obviously some of that is the time to analyze the data and then go back. But yeah, definitely acquiring the skill it requires some effort for sure. Dino, same question again, really sort of economic and technical challenges that you're currently encountering.
Yeah, so when we talk to customers, for example, that are doing antibody discovery using a single cell sequencing approach, one of the biggest issues is you can get a lot more sequences using these types, like a 10x chromium workflow, let's say, and some type of antigen baiting. However, when you get all of these sequences, you can't re-express all of them to test all those antibodies, right? So that becomes, because that's an economic issue, right? You have, let's say if you have 10,000, not 10,000, say 1,000 sequences, you're not going to re-express all 1,000 sequences to test their function. And so really where we see that there can be, where we need to grow and have new tools is having more insights on those sequences, on that information. So this is, again, connecting to like function of those antibodies when you're doing this single cell assays. And this connects to techniques we call seq-seq, secretion encoded single cell sequencing. I know we've worked with Revvity on using some of their oligo-barcoded antibodies for this, so that we can also look at how much binding there was to a target cell, what was the impact on the target cell's transcriptome, and link that to the antibody sequence. And so now you have information that gives you a prioritization that allows you to have more insight into this data and says, okay, I only want to look at these top 100 sequences and re-express those and analyze those. So, but one of the biggest challenges we're seeing is the kind of data overload where you can't, if you don't have insight into that data, you can't make the next steps in a reasoned way, and that allows, and there's a cost issue there, right?
Yep, I agree. And by the way, we really love collaborating with you. It's always a great time. So.
We do all sorts of crazy things.
So Nick, sort of same question to you. Just your experience with, the challenges that your collaborators are having, and both economic and technical, and how you work with them to overcome.
Yeah. Okay. So I'm incredibly lucky. We have a computational group which is embedded in my team, so we're able to help people right from the cells all the way through to the computational biology side, which is incredibly lucky, I know. And I've had that at my previous job with ICR and here, and it was really nice to come to an environment where we were able to have that. That's actually, in some ways, slingshot us into single cell and spatial based around pass and scale, etc. Other things that I can't talk about with NDAs in place, but later hopefully we can. I'm going to give you an example of where we've had problems and how much they've cost. So it's a real case that we've been involved with, and it's moving away from the analysis side, because we know that's a huge bottleneck. When I first came to Imperial, we had a senior PI project in place, which I inherited. Unfortunately, my focus was on getting funding and the quinquennial for our next 5 years, and I took my eye off the ball. We did a number of runs on the controller, 10x chromium controller, and we spent about 10 to 15,000 pounds, which is not insignificant amount of money. And money becomes blasé in when we discuss things like this, because that's how much things cost. But if someone put 15,000 pounds in my account, I'd be very happy. But when we talk about we spend it on projects, it seems to just disappear. So there is an issue with costs, and costs we can discuss in a second. But my team came to me and said, well, I think this has all failed, Nick, because we found 8 cells of his interest. And I went, oh, nuts. And we got the senior PI and his postdoc in, and we sat down and we said, I'm really sorry, we've only found 8 cells. And I thought, he would be rather upset spending all of that money. He jumped up and down with joy. First time I've ever been able to capture these cells, incredibly difficult to capture. And I was sort of sitting there with my mouth open thinking, well, what can you do with 8 cells? And I asked him the question, why? What? Again, I wasn't involved in the setup of this project. Why are you using 10x? And he went, oh, because I've read papers and everyone uses 10x. I went, no, come with me. So I took him into the lab and we looked at the cell M1. And I went, this one you can look at really rare cell types by surface cell markers, morphology, bright field, etc. You can put dyes in there for live or dead or dying. And you can train it to pick those cell types. So that was a 3-year study. That first 6-ish months doing the single cell 10x work to try and make it work. And the next 3 to 4 months was getting it working on there. We were capturing 60 to 80 cells, which was far exceeding what he was expecting. That 3-year project finished after that. So we finished this project incredibly quick, so we could focus on other things which come from that, from the computational biology side. So it is somewhat horses for courses. You need to understand the technologies, so you need to spend, someone brought it up earlier, you're there for 4 months just trying to discuss with them how. We don't take 4 months to do that, but we have a number of meetings with them to just discuss, where is the correct way to go? How much is this going to cost? What happens if it goes wrong? So it mitigates some of those problems. Risk management, who will pay for it if it fails, because this is a interesting project. And some of the ways that we've able to mitigate these costs is find out, buying the new IX system from 10x to be able to say use fixed cells. And I know Pass and Scale can use fixed cells, and effectively that's what they do. But we were looking at ways to, okay, maybe we should look at other ways to mitigate this when people can't fix them or don't want to fix them. And we looked, and there's a reason why we found fixing difficult. We'll come to that in a second. And it's based around costs and some of the caveats to looking at fixed cells. And we started looking at other technologies, and with the one we picked initially and what we still use is a Lavitas system that allows us to pick those cells. Now we can discuss bias, which is based around positive enrichment of cells. But if you have a poor cell viability score and you have to do filtering anyway, you try and increase those. You do increase your bias. Some of those are less robust cell types will not survive. But we found with the Lavitas system, it actually does. So based on fixed cells, we're looking at fixed cells now. We're going to go into the Pass and the Scale field, and also on IIX. That's the reason why we bought it. But we're finding it incredibly difficult to visualize some of these cell types on our current systems. So we're actually looking at the purchasing an Ascend from Revvity to try and help us with visualization. It's one of the better ones we've found. I think this is why I'm on this call, perhaps. And that's really helped us sort of figure out the cell numbers, because one of the complaints that we were getting when we were working with fixed cells is we weren't hitting that 10K cells or 20K cells, and obviously they're paying for that, and they're paying for the sequencing, so they want to get their funds, they want to get their bang for their buck, if you will. So effectively it's managing all of that, balancing all of that risk, and trying to mitigate some of it, and effectively that's a big chunk of my time, and the other chunk of my time is finding funds, but or funding and grants, etc. So yeah, that's I think I'll stop there.
Great, thanks. Yeah, it's interesting to me. I always think in terms of there's rare cells. So you have a population of cells, and you're interested in 0.1%, 0.1% less of them, and then there's another concept that you have precious samples, so you have very few cells. So it's not a needle in a haystack. It's just a few cells. You get what you get, whether it's a biopsy or whatever it is coming out of an experiment that you're doing. So real quick, we can run around with everybody. One, how do you approach the different? So one, do you work with rare cells and precious samples, and how are your approaches different for each one of those? I think Dino, let's start with Dino to go first. I'm trying to Robin it, and I'm forgetting who I asked to go first.
We do work with rare cells, so I'll just give you a few examples of different projects. One is in antigen-specific T cell identification and sequencing, and so we'll start with maybe 20 million PBMCs here. Obviously, 20 million PBMCs can't go through any of the instruments right now. So what we do is we actually use what we call nanovials. These are particles that have peptide MHC monomers on them, and we barcode those nanovials with streptavidin that's oligo-barcoded. Also, we get those from Revvity, so that we know which peptide MHC is on that particular nanovial. And then we fish out the antigen-specific cells. So that allows us to enrich that population of rare cells, and then we'll have tens of thousands. And actually, so we fish them out, but binding alone can lead to non-specific signal, right? So you can get things that will bind that are not specific, that are not antigen specific. And so we actually then look at how these peptide MHC molecules stimulate those T cells to secrete cytokines. We capture those cytokines also on the nanovial, and we tag those. And then we sort out that population to go into a single cell workflow. So that's that workflow then allows us to go from 20 million PBMCs to, it could be hundreds, could be 1,000 or so antigen-specific T cells with much higher fidelity, because we have they bind and they are activated to secrete. We use similar types of workflows for other rare populations, like plasma cells that secrete antibodies that are antigen specific. So we'll usually have an upstream processing step where we're using flow cytometry to enrich a population for the downstream use in the single cell sequencing workflows. So the other question is on cells that are rare, not rare, but that are scarce, I guess. So you have a scarce population from a tumor or from a biopsy. So this is really we see in any of the workflows and that you would get loss of cells. So you'll get whether it's dead volumes or other things, cells are sticky. We can, on average, we're losing about half of our cells as we go through any of these workflows. I haven't seen any workflows that are better than that. I'd love to hear from the other panel members on if there are any workflows where you don't lose cells. But we kind of have that as our acceptable loss. And so we have to then understand how many cells we need to start with in order to get to that level of cells that you're going to be able to make statistically relevant decisions on. So if you need 1,000 cells in the end, you're going to have to start with 2,000, et cetera. So that's how we kind of look at that. And if we find that we're not getting that many cells to start with from a sample, and going through all the processing steps of that sample, then we will kind of assume that that's just not going to work in a workflow.
Thank you. And I'm trying to go on mute as there's planes flying overhead, so apologize for that. So same question to you, Linda. How do you manage precious and rare samples profile? How do you manage profiling rare and precious samples?
Yeah, and some of it comes back to a couple things that Nick mentioned in his previous comments. I laughed really hard when he mentioned lab people coming in and saying, oh, I'm doing this because I read it in another paper. And I've heard that more times than I can count, and it always drives me nuts. I know that we, always have to have our references and our sources and stuff, but with the advancement in these technologies and how quickly it's changing, it's of course really difficult for, all of these individual researchers to keep up on every technology that's currently available. And that's why these conversations become so important. One of the things in terms of, rare sample sizes, is utilizing something like a combination of the Cell-in-One with a BioScribe platform. So being able to look at whole genome sequencing combined with whole transcriptome on individual cells, but taking those through a relatively gentle sorting protocol to be able to target the cells that are of interest. Or if you have things, needle biopsies, embryos, things along those lines, all of these things that where we can't even quite get through to sorting, and you just have these individual cells that you're looking at. We're really excited about moving forward with those bioscribe methods to allow you to get a really more complete picture and a more accurate picture of the genome specifically, and then coupling that together with the RNA expression. The other side of that is I'm always focused on ensuring the best approach for sample collection. And I'm not a huge fan of cell sorting. The concept of shooting your cell through the sorter and causing this added stress, and the time that is incorporated in that all leads to transcriptional changes. So one of the things that I like to push are things like the Levitas system, where we're using gentle levitation, and you can combine that with selection, positive selection, negative selection, to target those rarer cell types that you're looking for, but without putting it through a lot of stress that might lead to downstream impacts on the actual data, because it doesn't necessarily do you a whole lot of good if you've captured the cells that you're looking for. You've enriched for that, but you're getting kind of off-target results because of the preparation that goes through all of this. So those are kind of the major highlights for what we encourage when we have either really small samples, or when we're looking at things that you're trying to get through sorting. Of course, we have plenty of clients who will do a standard, flow sort and collect to do those more enriched. But, we still have to keep in mind the fact that, like you say, we're getting 60 to 70% recovery, best case scenario, on a 10x assay. I'm not a huge fan of the 10x fixed protocols. We've seen that, you've got to go into this with 1 million cells to maybe get out 10,000 in the end. And it's, it can be quite wasteful. Whereas a parse protocol, yes, the general capture rate is lower, but you can start with a lot fewer cells, and coupling that with the fixation. So it is really, the question always starts with what is your sample availability? How many cells do you have access to, and what do we need in the end? And it really guides that conversation as to what's going to come next. I've had clients working with pancreatic cancer cell lines that these cells can grow up to 200 microns in diameter. Well, we're kind of out of luck on a 10x instrument trying to do anything with that. They can't even sort these cells because they're so delicate that they basically just rupture during the sorting process. So, it is a manual pipetting cell collection to pull up a single cell and place it in a single tube to work downstream with a bioscribe protocol that then allows them to look at, you see these, 8+ ploidy DNA content within these samples because they just keep replicating without ever dividing. And it's a rare population. It's a difficult population to work with that really does not work even on, we looked at being potentially able to plate those using a cell in one. And even that was going to be too stressful on these cells. So it comes back to what we were talking about before. There's a lot of variables that can directly impact, the ability to even apply these samples to a particular technology.
I just had a follow-up, and this is a great point about flow and damage that can happen during the flow sorting process. So that's something that we actually were looking into, and how we could mitigate that using these nanovials that Partillion produces. And so nanovials are basically hydrogel particles that have a cavity. And so when a cell is inside that cavity, it's actually protected from the shear stress of flow cytometry. And what we actually see is improved viability, and we see actually differences in gene expression, especially for adherent cells, because these nanovials have an extracellular matrix, so that the adherent cell is sitting in this on this extracellular matrix, protected from shear stress as it's being sorted. And so that what we see when we then do downstream sequencing of those cells versus these adherent cells that are brought into suspension, like maybe these tumor cells you were mentioning, we see very different gene expression profiles. The ones in suspension do not have growth genes and cell division genes that are being activated, while the ones that are in adhered form on the nanovials actually do. And so this might be something where, where the types of single cell analysis, we bring everything into suspension, right, to do usually the single cell sequencing, but that might not be the case, or that might not give us the best results in terms of what gene expression is actually happening for adherent cells, in particular tumor cells, things like that. So having some type of carrier that can provide that environment for the cell, more like what an in vivo environment might look like with extracellular matrix, could help in some of those workflows.
Thank you. Very informative. Nick, commentary on precious versus rare sample types or rare cells?
I think that's been. I'd like to say some more, but we should be talking. Everyone else has said has just covered.
It, yeah.
It's been fantastic. I mean, it's nice to see that people are going in the same direction as us. I suppose all I'll add to that is just one has to become an expert in multiple different technologies to actually be able to sit down and work through this specific project with the PI or postdoc or PhD student, etc. And I think that's been the difficult thing, is being able to keep up. So I think if we can keep up, and we are the experts in those fields, hopefully one would, or you could argue this point, we'd be the ones with the best knowledge to actually pinpoint what technology we need to go on to actually get the best results for rare cell types or low numbers of cells.
Yep. Great.
I'll just add that.
Thank you. So I would like to take a minute. A couple questions have popped into the Q&A box. So just to remind everybody that if you'd like to ask a question, by all means, all you got to do is type it into the Q&A box and then hit send. And actually, this it's a nice segue. One of the questions is a nice segue into the next topic. Really, I'd like to, single cell. I mean, if we think about single cell NGS, it really started out very focused on transcriptomics, and now we've moved quickly into single cell multiomics, and so protein, obviously TotalSeq that I work with at Revvity, but then there's also epigenetic, right, doing ATAC, there's methylation out there, as well as, looking at spatial as well. So I'd like to just start and ask sort of your experience and advice to the people on the meeting, your approaches to working with multi-omics and thoughts and considerations. So maybe we'll start with you, Nick.
Yeah. Okay. Huge part of the lab is based around multi-omics now. I think that's just the future of the field, right? Yeah. That's where we're going to be moving to. I think even if it's us dragging us, screaming and shouting, that is the way that we're going to go forward to actually understand this. I'm incredibly lucky again. I sit right next door to proteomics. We work really closely with proteomics. We have linked projects, so we can do proteomic-based studies and genomic-based studies. One comment you made, my first endeavor into single cell was actually with DNA and genomically based and not transcriptionally based. So I sort of see that as something that is partly missed here, although ATAC does.
I would agree very much.
So I'd like to start looking at some of that. We're looking at methylation. When I was at Sanger, I was very luckily involved with Stefan Beck, who was the methylation king as far as I'm concerned. We effectively finished the CPG islands and where everyone was just joining them together. This is the original genome project, by the way. everyone else is just forcing them together as another repeat. We were actually sequencing them to make sure that we got something interesting out of it. And that was my first endeavor into, very early on in my career, into epigenetics, and that's where my love is. So it's for me, it's great to see spatial and single cell moving into that environment. But I think with me, I'd like to expand that even further, and that's the proteomics side. And I know some of the spatial technologies are now looking at the proteomics in single cell resolution. It's not the whole proteomics, only a subset of them. So I think that's something that we want to sort of really sort of foster in Imperial and becoming cutting edge in. There are caveats to all of this, and it's cell types and making sure the cells are good quality, and it harks back onto a lot of the stuff we've already spoken to in working with myometrium, for instance, in preterm birth that we did with Chelsea Westminster Hospital. I mean, they're incredibly large cells, very fragile. So what do you do there? Do you put them into the PAR system or scale, or do you just do nuclei? In the early days, we went to nuclei. We're now thinking maybe we can do spatial on those in single cell resolution to actually push that project further on and get more information. But then you've got to start looking at, okay, what transcripts are you capturing in spatial versus single cell? Are you capturing more? So there's lots and lots of areas where we have to normalize and make sure that we're capturing the right things. And I think that we're only just at the cusp of understanding all of this, normalizations and trying to get an understanding, that the same understanding we have with arrays and same understanding we have with qPCR. If back in the early days when qPCR didn't have any standards, and you can make them say whatever you wanted. In some ways, you can make spatial and single cell say whatever you want to as well. So having those standards in place is really important, I think.
Great, thank you. Linda.
Yeah, so I did see the question in the chat specifically about the 10x multiome assay. So I do want to chime in on that. And the comment was regarding seeing poor quality gene expression and ATAC results despite going into the assay with high quality samples. I am not the least bit surprised. It has gotten to the point where I discourage people who come in with the idea to run 10x multiome and say you are probably better off running a gene expression assay and a parallel ATAC assay because by trying to combine both together, the protocol results in sacrificing data quality on both sides. Generally, what I've seen in multiple labs is that you can get away with good gene expression and you don't necessarily get the high quality ATAC, or vice versa. You tend to get one or the other. And in fact, I worked with a lab when I was at Hopkins over the course of almost a year because we were seeing exactly that. Samples were coming in, they were 90% viability going into nuclei isolation. Excuse me. And the nuclei looked great. There are these little pinpoint perfect circles, and there's nothing that's going to indicate that we are going to run into a problem in the end. And we had one round where the ATAC peaks on the fragment analyzer, everything looks great, all signs point to yes, and we get data back in the end, and the correlation between gene expression and ATAC was just non-existent. There was nothing to be done with the data. And there were some technical issues on a product side that we were able to figure out and mitigate, and then run it again, and we saw the same basic results. And so it is not surprising to me that that particular assay struggles. And like I said, I tend to kind of lean away from it. In terms of the general concept of multi-omic approach, it's exactly where we need to be. The concept of getting as much out of a sample, out of a tissue as possible is exactly what we want. And it all comes from these new applications, combining something like a curio approach, a curio tracker approach, where we're getting true single cell or single nuclei resolution, plus spatial context, and then being able to incorporate things like, site-seq along with that. Even going a step further, taking a targeted xenium panel and moving that into also coupling it with a whole transcriptome Visium assay on the exact same tissue. I did and this is why I say about not just using, I read it in somebody else's paper as a rationale for something. A course that I taught last year, like the last class that we did, we sat down with a publication and I said, this was published a year ago. Based on what we've learned in terms of what single cell and spatial transcriptomics have to offer, if you were approaching this project now, what would you do differently? And, there were H&E images from a section, and then there was transcriptomic data from a section, and then there was, IF data from another section. And all of this is great, and it provides a really good picture. But when you can then condense all of that and provide all of that information from the same exact section and be able to look on all of these different levels and understand, the RNA, the DNA, the protein, the morphology within one section, I think it opens the door to advance it far more rapidly. I'm really excited because, Element is, we talk about sequencing, but we look at, the new Element of ED24. They're bringing out this cytoprofiling where the concept of being able to culture your cells on a flow cell and then look at RNA, DNA, protein, cell morphology within that sample. I think that's the kind of stuff that really changes our approach, that allows us to move into the next step of, true diagnostics and, clinical applications and things along those lines. So, those early stages were great, but, I always got the question of, well, how do I look at intracellular instead of intercellular? And, the original days of, of CiteSeq, now being able to look at both sides of things, I think we're really starting to hit that, exponential ramp up phase where multiomics is going to be a standard approach rather than the thing that if you could afford, you added in later.
Great, thank you. Dino, how are your thoughts on multi-omics?
Yeah, I think definitely a lot has been said already, and I agree with a lot of the discussion. I think as I laid out at the beginning, the layer that we are the most excited about is this functional layer and bringing that connection to transcriptomics and sequence. And so the way that we've been doing that is by looking at particular types of functions, and it could be physical functions of cells, morphology or contractility, but in particular looking at secretions, right? So one of the key functions, you think about cells, how they interact with their environment, what they secrete is how they broadcast information to their environment. And so, and there are a number of cells that you can't really define based on surface markers, but if you what they're secreting, you can define them very well. And so that layer of multiomics where you're adding this secretion profile of a cell along with transcriptomics, and this is that seq technique that I mentioned, that has been an area that I think is there's just so much to do. There's over thousands. There's like 2,000 or 3,000. I think it's 3,000 secreted proteins that are known to be secreted in the human genome. That's similar to the amount of cell surface proteins, but we really don't look at those proteins. We don't measure them at the single cell level. And so that to me is a huge frontier. Lots of new science is going to come out of that.
Great, thank you. So I would just like to take a second. So we have some more questions, and if you would like to ask a question, again, all you got to do is pop it into that Q&A box, and we will and hit send, and then we can start and answer those. So we've already taken the first one, so thanks, Linda, for answering that. I do have another one that is asking, when do you recommend nuclei versus whole cells, and how do you address protocols for digesting from solid tissues? And I guess we could throw that out for anyone to take.
I'm happy to
PhDs there. I'm going to back off and let someone else answer.
No, go ahead. I can follow up. Okay. So yeah. So that's a common question. And it's, what is it? Every bioinformaticist's favorite question or favorite answer is it depends. So that tends to, I think, fall a lot into the single cell realm as well. It comes down to, what's required, is usually, kind of how I approach it. There are options nowadays, better improvements for stabilizing tissue, that allow you to collect tissue in the middle of the night, put it in a storage solution, come back the next day, do your dissociation, and have whole cells rather than the need to flash freeze. So if you have the ability to do something like that, the goal is always to do whole cell because you want that more complete picture. You know that generally you're going to lose information by doing nuclei. But I've had labs who have run the gamut on all of it. They know that they're losing data, but the data that they're losing is irrelevant to what they're studying. So they're totally fine with collecting nuclei and just running the nuclei. We've had labs where their tissue their cells are not stable enough to undergo even a gentle dissociation, and they end up losing all the cell types of interest. So the only thing that works for them ends up being nuclei. So a lot of it tends to really just depend on the sample types that you're working with, as well as even always coming back down to budget. Is it a situation where can you afford to run one sample at a time every time you collect a piece of tissue? Or is it, you really need to batch and do something like a highly multiplexed approach like parse? All of those things come to play, and there's sacrifices, I think, that get made because of the budget. I laugh because when I first started doing this, everybody came in, you don't need biological replicates. Every cell is a replicate. And then miraculously prices come down and all of a sudden we need biological replicates. And it's just the general understanding of, we know that there's give and take on all of this. So whenever feasible, I always encourage trying to do whole cell. Working on methods to stabilize those cells. What does cryopreservation do to your cell types? So recognizing the limitations there, but so that's why I say it's a bit of it depends situation that really comes down to all of those factors that we've already discussed.
Great, thanks. And Nick, you had some comments to follow as well.
So yeah, it's horses for courses, unfortunately. What we've done on the top of what was just said, because that's effectively covered everything that I was going to say again, which is fab. We have, I'm going to a big shout out for my team. Again, I'm incredibly lucky. My team are willing to travel and willing to sort of go the extra mile. And we've actually sent our controller, we've boxed it up, sent our controller to a different lab within Imperial or with one of our partners to be able to do it in situ. So and it might be one sample. We've discussed this, and there's a cost implication of doing one sample around the plasticware. This is with 10x Visium. With 10x controller workflows. So single cell. And we've done it in situ. So we've been at that lab, they've done the workflow, we've helped them do that, we've done the QC, and we put it on straight away to stop that cell death as much as we possibly can. And I think that's sometimes what you have to do to get the best results or get the bang for your buck, if you will. And sometimes they have to realize that they may have to pay a little bit more for the plasticware because it's only one sample at a time. So we've done that. And that actually has saved a number of projects. Another area which saved a number of projects is our Lavitas system. It's literally saved. It's probably paid for itself over 10 times now with how many projects it's saved by just clearing out those dead cell side. And I fully understand and I fully agree with the fact if I can do it, I'll do live cell every single time. I think that's an area that I'd like to push, and hence me sending the machine elsewhere. And I think everyone else has said, it's been said, in the previous discussion, what I wanted to say. So that's where I'm going to stop.
Okay, great. So I know we're coming on time with about 5 minutes left. I do want to give you guys a quick opportunity to answer a little more higher level question, but how do you see single cell sequencing as well as spatial transcriptomics contribute to personalized medicine over the next 5 to 10 years? And who would like to start first?
So I can start here. So I think that where I think that there's a number of points, and we'll probably hear about kind of characterizing tumor tissues and other things. One of the areas that there's so much excitement because there's so much variability person to person is in our immune system, right? So if we look at the repertoire of T cell receptors, if we look at the repertoire of our B cell receptors, right, of our antibodies that we're producing, all that is so unique to each individual and so useful potentially, and it's a chicken and the egg problem, and it's a cost problem. But there are studies that are starting now. That one is the Human Immunome Project, and maybe some of you are familiar with this. This is looking at 10,000 patients, looking at huge quantities of different markers, biomarkers of the immune system. Some of those will be single cell markers. And I think that once we have more information on how that can presage disease, so whether we see unique T cell receptors that are and antibodies that are presaging type 1 diabetes, other autoimmune conditions, et cetera, I think all that then starts to play a huge role as we think about precision medicine, right? Because that is now defined for that individual. And we could also look at how that individual is changing over time. How is their repertoire changing as they evolve over their lifetime? And how is that going to be predictive of disease? And ideally, precision medicine, one way that people have looked at precision medicine, you have this mutation in your tumor cells, and I'm going to give you this particular drug. That's almost too late. That's at the point where you're saving, 6 months to a year, 2 years. Where I think the big advantage is, where we can have the most impact, is if we're intervening at the earliest stages, where we're looking at, preventing tumors from forming by maybe looking at the immune cells that are responding to and killing those tumors, or the lack of those immune cells in certain patients. So that's my kind of pitch there, is that I think that one of the key areas is if we can lower the cost still, but be able to monitor our immune system at that really single cell resolution at scale.
Thank you. See, Nick, would you like to add?
So T cell, B cell is definitely an area which we need to expand into. And it wasn't actually the person who asked the question, I think it's Laura. I can't really see it very well. That wasn't added into her question, but it really should have been. And we're working in single cell and so on and so forth. But to add to that, it already is. It's clear. We're working with clinicians where they have samples that come from the clinic. We did a tissue array in spatial to try and reduce the costs. That tissue array had X amount of spots, and I can't remember how many right now. Apologies. We ran that, and we did a load of experiments based around, analysis based around those samples, and the clinician looked at the data and went, wow, I can actually pick out my different patients from where they're in treatment and where they're not in treatment based on their transcriptional values and what they look like. And we're now drilling that for biomarkers for this. It's a fibrotic lung disease that we're working on in the time. The key point here is they cost about £100 per spot. That's an incredibly useful technique at not a lot of money. I don't know how many dollars that is. Apologies. Maybe someone can do that and stick it in the chat. But that is still quite cheap in terms of in terms of a diagnostic test, and that's the reason why when we discussed earlier, I said we are looking at the pathology side already. And so I think we are going to be looking, and we are already touching on personalized medicine.
Thank you. So with about a minute left, Linda, any added comments?
Yeah, no, I mean, it's like I just think about the kind of alphabet soup that we talk about when it comes to spatial, you know, TMEs and TILs and TLSs and all of these things that are so predictive of, you know, what the treatment is going to be, what the survival rate is going to be, and all of that comes from understanding, you know, combining that, you know, importance of the immune system with that spatial context. And the same thing, you know, taking a Xenium slide and being able to put half a dozen, you know, tumor sections on there, and getting to a point where we can like kind of mass produce this diagnostic approach is where, you know, we're certainly at Omapics, what, you know, we want to push forward and bring this into kind of an everyday routine, as opposed to, you know, waiting for, you know, months to know how a person's going to respond. You know, have that profile that tells us that early on. So as that quick summary.
Great. Well, thank you very much. So we're definitely at time. I'd just like to thank the three of you so much for your time. It's been a great discussion, really valuable insights. So greatly appreciate that. Also, before we go, I'd like to thank the audience. Thank you for your questions. And just to let everybody know, this webcast can be viewed on demand. Lab Roots will send an email when it's available. And also, we have a resources section around single-cell sequencing, a single-cell infographic, as well as my group's latest product launch, which is a PhenoCyte kit for profiling protein at large scale that we co-designed with Scale Biosciences. So again, thank you very much for the time. Dino, is always great to see you. Linda and Nick, it was wonderful to meet you. And thank you everybody for joining.